Hello, everyone. Warm welcome to all of you. So thanks to all of you to attend this webinar on IVDR performance evaluation. Just a few points before we get started. First of all, this webinar will be recorded and the recording will then be available. So that's something to keep in mind. And second of all, we will have a Q&A session at the end of the presentation. So for that, please ask away any of your questions in the Q&A section on the bottom right-hand side of your screen. So in the Zoom app, you have a Q&A section there, and you can ask all of your questions, and we will be able to see it at the end of the presentation, and then we can initiate the Q&A session based on that. Just for your information, you can ask the questions anonymously, but keep in mind that if there are too many questions, we may not be able to review all of them. So if you want to make sure that we follow up on your questions, please ask them with your full name, and then we will get back to you if we cannot review it during the Q&A session. So that's the first 2 points. Just before getting started, I will just quickly, briefly introduce the company that is hosting this webinar, Product Life Group. So it's just a quick overview to know who is talking to you and basically what we can provide as a support. So Product Life Group is a global one-stop shop for all things compliance related to healthcare industries. We can support you with a fully integrated expertise and a global reach. Our model is based on a scalable partnership, so we support early stage startups to large groups. Basically, we can support you for global strategies, but also local projects. So that's why we have various offices all around the world. As you might have noticed from my slight accent, French is one of them, but don't worry, we'll try to keep our English clear and concise all along this webinar. If you can go on to the next slide. If we look at in vitro diagnostics specifically, we can support you in all things related to quality, so creation and maintenance of quality management systems for pretty much any economic operator. We can also support you in terms of regulatory affairs throughout the entire life cycle of your product. So from the very early stage of design and development, so the very first spark up until the post-market activities. We can also support you, and that's actually the topic of today, in the clinical aspect. So from the strategy to support for evaluations, including performance evaluation and investigations. And also we can support you in terms of market access for the strategy and support for access and reimbursement. Basically, our goal is not to only advise you, but we can also help you execute. We can provide operational support so that if that's something that you're looking for, at the end of the day, we can also support you on that. And then next slide, please. And we can support you all along the life cycle of your product. So from the very first spark, so early design and development projects, throughout the conformity assessment of your product, including, for instance, performance evaluation, which is part of our topic today, all the way to post-market activities. Our goal is basically for you to have one partner that can help you accelerate your medtech success every step of the way. So with all that being said, I know that you're all here for the performance evaluation of in vitro diagnostics. So I'm very happy to welcome my esteemed colleagues, Laura Panico, who is regulatory senior consultant, and Jordan Delille, who is a clinical manager consultant within the Product Life Group. So guys, the floor is yours.
Thank you,
yes,
Samuel.
thank you. Thank you, Samuel, for the introduction. And hello, everyone. So this webinar will be in 3 parts. An introduction to the notion of performance evaluation. The second part will develop how to demonstrate the performance of an IVD. And the final part will focus on RUOs and in-house devices. So first, introduction to performance evaluation. Before an IVD can be placed on the market, the manufacturer must affix the CE mark to its device. And to do so, they must demonstrate compliance with the requirements of European regulation. In particular, he must provide clinical evidence that the device is safe and achieves the expected clinical benefits with reference to the state of the art in medicine. In other words, in addition to demonstrating the safety of the product, the manufacturer must scientifically demonstrate the positive effect of the device due to its function, such as screening, monitoring, diagnosing, or assisting in the diagnosis of patients, or the positive effect on patient management in terms of public health. This is the definition of the clinical benefits in the Article 2.37. If we refer to the definition of clinical evidence given in Article 2.36, it is the clinical data and the results of the performance evaluation which make it possible to assess whether the device is safe and whether it offers expected clinical benefits when used in accordance with the manufacturer's intended purpose. So the performance evaluation is therefore an essential element to obtaining CE marking for an IVD. So, okay, I'm a manufacturer. I need to realize a performance evaluation of my product, but what it is? Article 2 gives a first definition. It is an assessment and analysis of data to establish or verify the scientific validity, the analytical, and, where applicable, the clinical performance of a device. So we understand that there are several components. The Article 5 precises that it is necessary to demonstrate the conformity with general safety and performance requirements, the GSPRs. And finally, Article 56 says that it provides sufficient clinical evidence. So in short, to demonstrate that a product is safe and produces the expected clinical benefit, the manufacturer must provide clinical evidence. This clinical evidence is based on the three pillars of performance evaluation: scientific validity, analytical performance, and clinical performance. To understand the importance of these three pillars, we need to go back to the definition of an in vitro diagnostic medical device. So what is an IVD? How does it work? It can be defined by its function, like screening, diagnosis, monitoring, prognosis, etc. The method, type of technology, whether it's automatic or manual. The type of data, is it qualitative, quantitative, or semi-quantitative? What type of user is it intended for? The patient himself, a healthcare professional, trained personnel? And in what environment will the test be performed? A lab, a patient hospital bed, at home, in an ambulance, maybe? The input data for this device are the target population, from which the samples will be collected, the samples in which the analyte will be measured, detected, or quantified. It can be blood, serum, cells, urine. The analyte measured, quantified, or detected. It can be a biomarker, a chemical component, an enzymatic activity, whatever. And in case of companion diagnostic, the drug associated with it is also an input data, of course. And as output data, the analysis by the device will produce information concerning a clinical condition or a physiological state, sorry. So let's make a focus on the three pillars of performance evaluation. Now that the product is clearly defined, okay, let's return to the definition of the three pillars. So the first one corresponds to scientific validity. This is a new feature of the regulation which did not exist under the directive. Scientific validity is defined as the association of an analyte with a clinical condition or physiological state. It demonstrates the value of measuring, detecting, analyzing the analyte in question or its properties. Scientific validity is therefore the process of establishing a correlation between the analytes and the clinical objective of the IVD. In a way, scientific validity demonstrates how the device can have a positive impact on the patient health. It defines the expected clinical benefits that the medical device must achieve. In this picture, we can see that the data to be taken into account for the demonstration of scientific validity are the target population, the type of sample, the analyte, and the clinical condition or physiological state concerned. Scientific validity will only use these elements whatever the method, the environment, the type of data, the users. So we see that scientific validity can be demonstrated independently of the IVD. So we saw what parameters of an IVD are involved in the scientific validity. Let's do the same work for the second pillar, analytical performance. The analytical performance of a device represents its ability to correctly detect or measure a given analyte. The analytical performances are established by considering one or more sample types, the analyte, the method used, and the type of data generated. The users, the environment, and the clinical or physiological condition are not considered for analytical performance demonstration. Finally, the third pillar of performance evaluation, clinical performance, is the ability of a device to produce results correlated with a given clinical condition or physiological process or state, depending on the target population and the user for whom the device is intended. The IVD components mobilized to demonstrate these clinical performances are different from those mobilized to demonstrate analytical performances, since the IVD's clinical performance will be defined by considering the target population, the device's function, as well as the user and the environment in which the device is to be used, and the physiological state or clinical condition qualified by the device. In the case of companion diagnostic, as I said before, the drug associated with the companion diagnostic will, of course, also be taken into account for the clinical performance. So to establish clinical evidence that the device is safe and offers the expected benefits, you understand that we cannot simply demonstrate scientific validity, analytical performance, or clinical performance alone. It is a combination of all three components that enables us to cover all the parameters of the device and provide the data needed to demonstrate the clinical evidence. So let's make a first summary. We saw that performance evaluation is the assessment and analysis of data to establish or verify the scientific validity, analytical performance, and where appropriate, clinical performance of a device. It allows compliance with general safety and performance requirements to be demonstrated, and it provides scientific evidence that the device is safe and offers expected clinical benefits. We will now discuss how to demonstrate the different pillars of performance evaluation. First, the scientific validity. The demonstration of scientific validity is highly linked to the intended use of the device. And to illustrate these points, let's take the example of 3 devices, all designed to measure C-reactive protein, the CRP, but with different uses. In the first row of the table, let's imagine that the claim of the first device is to determine the level of CRP in the serum of patient to assess the inflammatory state of the body. So to demonstrate the scientific validity of this kit, we will need to show how CRP protein plays an essential role in the body's inflammatory response and under what circumstances this level is abnormally high. Moving on to the second line of the table, if we now consider a device whose purpose is to diagnose or confirm a pathological condition associated with an abnormal level of CRP, in the case of rheumatoid arthritis, for example, we will need to demonstrate that the abnormally high level of CRP in the blood is associated with rheumatoid arthritis. And finally, last example, let's imagine a third device whose claim would be to enable monitoring of CRP levels in patients treated with an anti-inflammatory drugs to reduce the inflammatory state and hence CRP levels. The manufacturer would have to provide proof that CRP levels are a good indicator of treatment efficacy and that they enable the physician to adjust the drug dose. So as we can see, the evidence required to demonstrate scientific validity of a device differs according to the manufacturer's claims for the device. The regulation specifies the sources to be used alone or in combination to demonstrate scientific validity. This will depend on the data available, of course. The association of an analyte with a clinical condition or physiological state is often documented by academic researchers or studies evaluating the potential of said analyte for a specific clinical application. Manufacturers can use this peer-reviewed scientific literature to provide evidence of scientific validity. Scientific literature means journal articles, conference posters, guidelines, or documents from official websites, such as the IVDR, the WHO, the EMA, etc. The manufacturer may also use expert consensus opinions from relevant professional associations. If there are other devices measuring the same analyte for the same purpose, whatever the method, the manufacturer may use the data available for these different devices to document scientific validity. And in the case of innovative analytes for which scientific validity is emerging, it's likely that the manufacturer will need to demonstrate this through feasibility studies or clinical performance studies. We will take a closer look and detail when to use which sources of clinical evidence. So first question to ask is there a consensus scientific validity is established? This is the case for the devices such as pregnancy tests, where we know that beta HCG is produced by the embryo shortly after conception, making it an ideal marker for detecting pregnancy. Another more recent example is PCR testing for detect people infected with the COVID virus. The scientific validity of S or N protein RNA is widely described in the literature by the WHO and even in the guideline MDCG 2021/21. And for these cases, there's no need for a systematic literature review. Reference to the guides or to a few reference documents in the field will be enough. The manufacturer can also use relevant information from other devices measuring the same analyte, but be careful, however, to quote references that corresponds to the intended use of the device, the target population, etc. If there is no consensus to validate the link between the analyte and the physiological or clinical state, a systematic search of the literature should be carried out to find evidence of the relevance of measuring this analyte for the intended use. Or identify other data, such as data on similar devices, design feasibility studies, or other clinical utility studies, for example. Once the data have been identified, their relevance to demonstrating the link between the analyte and the clinical condition needs to be appraised and analyzed. And by relevant, we mean data that demonstrate the link between the analyte and the clinical condition on the target population and corresponding to the device's destination. When there is no evidence of scientific validity, or when such evidence is deemed insufficient, or where the degree of novelty is high for new analytes or new intended use, it will be necessary to provide scientific justification and generate new or additional data. The manufacturer can determine what additional evidence is required by carrying out a gap analysis. He may then specifically design a study to demonstrate scientific validity, or if clinical performance data are available, may potentially reanalyze them to provide further evidence of scientific validity. So we've talked about the first pillar of performance evaluation, and now let's look at how to demonstrate the device's analytical performance. Demonstrating analytical performance means demonstrating the performance of the method used by the device and must answer the question, Does the device measure or detect the analytes reliably, accurately, and consistently? The relevant performance characteristics, which form part of the GSPRs and are linked to the analytical characteristics of the device, must be supported by existing evidence or by the generation of new evidence. In the vast majority of cases, this can be demonstrated through performance studies. The section 9.1 of Annex 1, which I invite you to consult, lists the analytical performance to be assessed by the manufacturer. In particular, the regulation mentions analytical sensitivity, precision, accuracy, analytical specificity, measurement range, linearity, etc. These indicators may depend on the test technology and the intended environment of use. The importance and weighting of the various indicators listed in this section should be considered on a case-by-case basis, and not all are necessarily applicable. However, any omission must be clearly explained and justified. The choice of a criteria to be evaluated in terms of analytical performance may be guided, among other things, by the results of the risk analysis. For example, measurement variability criteria to be taken into account, choice of interference to be tested, etc. Okay, let's turn now to clinical performance evaluation, which, as we've seen, involves demonstrating a device's ability to produce results that correlate with a particular clinical condition or physiological state in relation to the target population and intended users. The manufacturer should demonstrate that the IVD has been tested for the intended use, target population, use condition, operating in use environment, and with all the intended user group. Indicators of clinical performance vary and depend strongly on the intended purpose and performance claims. The IVDR sets out clinical performance may not be required for certain devices in Article 2.39. For example, clinical performance data may not be expected for non-sterile specimen receptacles, microscopic glass slides, or some general reagents. In such cases, and where due justification is given, a clinical performance report would not be expected. So the clinical performance of a device is demonstrated on the basis of one or more of the following sources: clinical performance studies, of course, peer-reviewed scientific literature, or published experience from routine diagnostic tests. And by published experience, we mean any document or data set resulting from the use of the device that has been the subject of a brochure aimed at the general public, a publication on a laboratory website, an internal document available on request, and officially announced information. In fact, all public information can be used as a published experience to demonstrate the clinical performances. So for the second take-home message, we can say that the three pillars of performance evaluation, that are scientific validity, analytical performance, and clinical performances. We saw that scientific validity depends on the intended use. It can be demonstrated independently of the device, and the sub-strategy have to be well described in the performance evaluation plan. For analytical performance, they are generally the same for all IVD, but may vary depending on the function and the technology of the test. And finally, the clinical performances depend on the intended use and the manufacturer's claims. So now I like the talk to Laura, who will talk about RUOs and in-house devices.
Thank you, Jordan, for this first part on performance evaluation. So let's see together what about performance evaluation for research use only devices and in-house devices. So let's start with research use only devices. So first of all, what are these kind of products? So there is no dedicated definition of research use only products. There is no definition in the regulation and no definition in the MDCG for now. But what is important to note about research use only products is that they are products intended solely for research purposes and not for diagnostic or therapeutic purposes. Here, the key point is, if you want to commercialize a research use only products, you cannot have some diagnostic claims about the device, and you cannot generate patient diagnostic results. So here are some examples of research use only products. So for example, antibodies sold for biomarker exploration, or ELISA kits for measuring a protein in research samples, qPCR instrument or reagents used in the research to study gene expression, or as part of software devices, you can have some bioinformatic tools for exploratory data analysis, and AI models trained on research datasets to identify biomarker patterns. And now, what are non-research use only products? So for example, a COVID-19 PCR test marketed for detecting infection in patients, and a software that analyzes sequencing data from a patient sample to diagnose cancer. So here you can find the difference between research use only products and non-research use only products is mainly based on the fact that research use only products cannot generate patient diagnostic results. And that is the key point here. So now we have defined what are RUO products. What about performance evaluations for this kind of products? So as they are not considered as IVDs under the IVDR, first of all, the intended purpose should be clear and should be for research use only. And the label on your device should mention for research use only. You must avoid diagnostic claims in labeling and promotion, because if there are some information in your claims, labeling and promotion, that can let some people think that your device makes some diagnostics, it can be reclassified as an IVD. And if your research use only device is used for diagnostic purposes, then it becomes an IVD, and it should be compliant to the IVDR. So for this kind of device, no performance evaluation is required because they do not fall under the scope of the regulation. But if your product is later intended to become an IVD, then performance evaluation requirements under the IVDR will apply. And you can make transition from research use only products to IVDs. But when you perform this kind of transition, you need to have a clear change of the intended use of your device. And it will show that now you're an IVD and you should follow the IVDR obligations. So what are the key messages regarding research use only? So it's important to understand that research use only products are not IVD devices. They are very different. The main difference is about the intended use, and it is the intended use of your product that defines its qualification under RUO or IVD. You need to have clear labeling and no diagnostic claims, because diagnostic claims are critical. And then performance evaluation does not apply to this kind of products. So now we will continue with in-house devices that are a little bit different of research use only devices. So what are the in-house devices? For this kind of devices, we have a clear definition. In-house devices are devices that are manufactured and used only within a health institution established in the Union and that meets all conditions set in Article 5.5 on the IVDR. They are developed to meet specific patient needs when no equivalent CE-marked IVDs are available. So, for example, in-house device can be a PCR master mix that is used within a health institutions. Or it can also be a software that is developed by a healthcare institution and that is used only on site by healthcare professionals to diagnose, for example, a rare disease. In parallel, non-in-house devices can be a medical device application where patient can use the application outside the health institution, or, for example, a COVID-19 self-test that is used outside the health institution. So what is important to note about in-house devices is that they are devices that cannot be used in the large scale. That kind of devices are used only within a health institutions. And that is the main point that differentiates them from IVD devices. So what is the regulation around the in-house devices? So in-house devices are well regulated by the IVDR and they are regulated under Article 5.5. They are exempt of CE marking, but there are some conditions that need to be met. The first condition is that these in-house devices should be manufactured and used only within the same health institution. They shall not be transferred to another legal entity. The laboratory that develops these in-house devices shall comply with EN ISO 15189 standard. There needs to be justification that no CE-marked IVD exists to replace this in-house device. You shall have the documentation that describes design, manufacture, and performance of the device. And the in-house devices shall comply with the GSPR. And this last point is very important because even if your in-house device is not CE marked at the end of the day, so you cannot affix a CE marking on your in-house device, it must comply with the GSPR. And what is the reflex of the GSPR as part of the IVDR is the technical documentation. And so for in-house devices, even if the technical documentation may be more light than for an IVD, you shall have a dedicated technical documentation for in-house devices. And then what about performance evaluation for these devices? So in-house devices must demonstrate performance, and they must demonstrate analytical performance and clinical performance too, because these are requirements from the GSPR. The main difference is that the evaluation should be proportionate and adapted to the fact that the device is used only within the health institution. For example, for an IVD device, sometimes within clinical performance, it is really helpful to go through multicentric studies to show that there are no difference within the center the device is used about the use of the device. But for an in-house device, multicentric study could be not appropriate because the device will only be used in one center. Okay, so that is some point that can be different between clinical performance studies for an IVD and clinical performance studies for an in-house device. So your effort should be proportionate to your product. For this kind of device, evidence must be available internally to demonstrate safety and performance. And that's why I told you before that you must have a technical documentation for this device. And specifically, you should have your GSPR checklists. And there will be no formal IVDR conformity assessment, but traceability and documentation are mandatory. So when you have in-house device, you will not be subject to an evaluation by a notified body, but you may have an evaluation, for example, from competent authorities as part of the evaluation of your laboratory, and because you have an in-house device. So what are the key messages for in-house devices? So in-house devices are exempt from CE marking, but are not unregulated. They must meet Article 5.5 conditions and relevant GSPRs. They need to have performance evaluation, but the level of performance evaluation should be adapted to the device and to its use in only one health institution. And if later commercialized or shared externally, this device will become subject to full IVDR compliance. So now, just a few words on conclusions about this webinar. So performance evaluation is really central to demonstrate safety, performance, and clinical benefits of IVDs. It's built on 3 pillars: scientific validity, analytical performance, and clinical performance. For research use only products, there are not IVDs. No performance evaluation is required. They should have clear labeling and no diagnostic claims. And transition to IVD will trigger full IVDR compliance. And regarding in-house devices, they are exempt from CE marking, but not unregulated. They must meet requirements of Article 5.5. They should demonstrate compliance with GSPR, and performance evaluation should be adapted to use within the health institution.
Thanks a lot, Laura and Jordan, for this very nice presentation. I want to give you a warm thanks from, I think, pretty much everybody in the assembly. Just as a reminder, if you have any questions pending, if you want to go further on any subject, then you can click on the Q&A part right under the screen. So you have a question mark with Q&A. You can write your question there, and then we will answer them. In the meantime, while we're waiting for your questions, I just want to briefly introduce some of the services that we can provide to you in terms of performance evaluation and broadly everything related to basically the evaluation pathway in the end, basically, of your design and development, and also in your IVDR transition. So we can definitely support you in everything related to performance evaluation strategy, the establishment of performance evaluation plans, and performance evaluation reports. And we can also support you in undergoing your clinical investigation protocols, for instance, for clinical performance evaluation data. Basically, we are quite flexible. We can adapt to your company. So we can have a project-based approach, a retainer-based approach. We can also dedicate a bucket of hours to your company, or we can even provide you with a staff augmentation for this purpose. So thanks a lot for everybody in attendance, and now we will go on to the questions. So let's get on to the questions. So I see a first question: can an in-house assay be used for samples from another institution?
So, sorry, Samuel, I didn't hear well the question.
The question is related to in-house devices and in-house assays. But
yeah.
It's related to those in-house assays be used, well, can they be used in from other institutions?
So basically, No, basically the principle.
Sorry, I didn't fully grasp the question. It's for
Sorry.
samples from other institutions. Basically, you have one institution that have samples, they send it to another institution, and the institution that has the in-house assay performs the test.
Yeah, absolutely. So yes, it is something that is visible. So you can collect samples in other health institutions and do the test only in the institution and send the results of the test to your other institutions. It's something that is feasible. The principle of in-house devices is that the assay and the test should be performed in only one health institutions, but the samples can come from other health institutions.
Okay, perfect. I have another question. Is it mandatory for the clinical performance to perform the clinical performance for products that are research use only? I think you touched a bit on that, but I want to have your opinion right now.
Sorry, can you repeat the question?
Is it mandatory to do the clinical performance evaluation for products that are labeled as research use only?
Okay, so as I said during the webinar, clinical performance evaluation is not a requirement for research use only products because they are dedicated for research and they do not fall under the scope of the IVDR. But if your device in the future aims to be transformed from a research use only device to an IVD, then clinical performance will be mandatory. But it will be before you will fall under the scope of the IVDR.
Okay, perfect. That's quite clear. Another question is related, so it's a bit in between clinical evaluation and regulatory affairs. Must the IFU present the average or the most relevant clinical performance data when several reference methods exist?
I think it depends on how many reference you are talking about. Sure that in the performance evaluation report you will have to explain all the reference methods you use and all the results. But if you mean in terms of IFU, for example, if you want to show only the comparison with the reference method, the gold standard, yes, you can only put the results of the comparison with the gold standard in the IFU, but you have to be you have to show all the reference you have in the performance evaluation reports.
Okay, thanks a lot. I have another question related to IVDR performance studies for home testing in vitro diagnostics. How do you handle it?
For home testing?
Yeah, so basically we are touching a lot more on what is used in the lab, but for like self-testing, home testing in vitro diagnostics, how do you perform the performance evaluation of these specific devices?
Maybe Jordan, you can handle this question.
Okay, sorry, I was reading another question on the chat.
I will take, no problem, I will take the point. So the question, Jordan, was about what about performance evaluation for home testing and self-testing? And for this kind of devices, performance evaluation is also applicable, and the 3 pillars are also applicable. So you should follow the same methodology as for any other IVD for these devices.
Yeah, and just to briefly touch on that topic, something to consider for your own testing in vitro diagnostics is that you need to take into account the risk associated with the handling of the in vitro diagnostic within the home setting. So that's definitely something to consider and to clearly evaluate within your clinical performance evaluation, but also your usability testing, usability engineering file. So that's definitely something that needs to be considered, that is on top of what you usually have in the lab.
Yes, and to complete the answer, yes, for the technical performance, you will do it for performance studies in lab. I mean, in your institution. But for the clinical performance, do not forget that you have to test your device in the environment of use. You have to use to assess the device in the real use, in the real world condition. So if it's a home testing device, you have to test it at home with patients.
Okay, perfect, quite clear. I have a question regarding research use only products. What can be shared or published in relation to the analytical performance of the research use only products?
Do not hesitate if my answer do not totally answer your question, but regarding analytical performance for research use only products, you can mention practically whatever you want. The thing that you should not forget is that the analytical performance you claim shall not let think external people that your products may perform diagnostics. So, for example, we draw down some kind of example of analytical performances, for example, sensitivity, etc. And if you talk about sensitivity only to detect something, it's okay. But if when you talk about it, you talk about sensitivity to detect something for the diagnosis of something, it will not work and it will fall under the IVD. So just be careful when you talk about your analytical performance for RUO products to not think that your product can be an IVD, and do not make any analytical performance claim that let think that your product may be an IVD and not only for research purposes.
Okay, so we have a quite specific question regarding next generation sequencing, which is a typical example of a research use only technology that is used in lab as an in vitro diagnostic. They fall under, therefore, the Article 5.5. How do you see performance evaluation challenges inherent to this technology as in-house, considering that there are thousands of analytes and combinations of analytes within the next generation sequencing field?
I don't think I will be able to answer this question as part of this webinar because this is really specific. Maybe the person who has this question may just add some details about what he or she intends to have as an answer, because
okay, perfect. As this was an anonymous question, can you please, if you send a follow-up question, can you please send it non-anonymously so that we can follow up on that? Thanks a lot. So I have a question regarding the number of sites on which the performance evaluation needs to be carried out. Does the clinical performance evaluation needs to be carried out on different sites, or is one enough?
Okay, it is not mandatory to have different sites. You will just introduce a bias if you only selected one center, because the way they analyze, etc. It can be specific to one site. So it depends of your claims on how many samples you want to analyze, and yes.
Okay. So it's really product dependent and project dependent from what I understand. So we will need to get into the specifics of the different clinical evaluation that the person is asking, right?
Yeah.
Okay. Are documents generated from, so we are talking about in-house testing. are the documents generating from one health institution, can they be transferred to another health institution that will perform in-house testing in the same way as the one that developed the documentation beforehand?
I think this is something that is feasible because it is documentation. But what is important to retain about in-house devices is that they are devices that are developed and used because there are no other device in the market. And if there is a transfer of documents from a health institutions to another, then it will be difficult to justify that the need is not covered by other devices. And maybe in the future, it will be difficult to handle this kind of transfer of documents because maybe the competent authority will just state, okay, so there are other health institutions that are doing the same things, so there is a need to create an IVD and you will have to switch to IVD. So be careful about that, but I think it may be feasible.
Okay, perfect. Yeah, I want to touch on that a bit more. Yes, what Laura said is completely true, and you can transfer at least partially the documentation, but please consider that the environment may not be the same, the people that are working in the institution may not be the same. So part of the documentation may need to evolve as well to be compliant with the regulation as well. The next question is actually related to what you just said, is basically the Article 5.5 mentions that the N-scale institution needs to justify in its documentation that the specific needs of the target patient group cannot be met by CE-IVD products of an appropriate level of performance. What does that practically mean in a real context?
So it means that if there is an existing IVDR product that do the same thing and that is available in the market, you cannot consider your in-house device, and you cannot consider your device an in-house device, and you will fall under the full scope of the IVDR. But this is something you have time of, because for now, as Udemy is not fully launched, there are no possibility for the actors to verify that the needs is really not met for the patients. So you have until 2030 for these requirements to justify that the need is not covered by an IVD device on the market.
Okay, quite clear for me. Next question is related to the transition of a research use only products to IVD. How long would it take to transition RUO to IVD, for example, a PCR assay for disease assessment? I think we will fall in the same type of answer for some other questions. That really depends, right?
Yeah, my answer would be it really depends. It really depends on how you work, on what is your structure. I cannot answer this question, but what you need to consider is that for your research use only products, basically, I think you do not have any clinical performance data, and it will take at a minimum the time to perform the clinical performance evaluation and to gather clinical performance data on the devices. So you will have to conduct, it is highly probable that you will have to conduct clinical performance study. According to the standards, you will have to submit it to the national competent authorities where you want to launch your study, to get the results of your study, to get the report. So it can take a little bit of time.
Yeah, And to touch further on that, on the more regulatory aspect, it will actually depend also on the amount of documentation that you have on your research use only products. If you have design and development documentation, manufacturing documentation, risk assessments, it actually quite depend on the amount of work that you need to do. It can be quite streamlined if you know where you're going and if you have adequate support or adequate persons within the company that can transition, but it will depend on a various amount of parameters. I fully noted your question regarding the NGS question. I think it would be best for us to initiate a meeting so that we can discuss the approach for this type of product, which require thousands of combinations, targets. It's actually quite specific, and there are strategies that are available to streamline this clinical performance evaluation to get onto the market. But yeah, it would be better to go more specific on this aspect.
Yeah. But we know that there are some issues for NGS manufacturers. Yeah, it is something that is difficult to handle with performance evaluation.
Okay, I don't see any further questions, so we're right on time. So thanks again, Laura and Jordan, for this nice presentation, this nice Q&A session. If you have any further questions, do not hesitate to send us a message, an email, so that we can follow up on the questions. If you didn't attend the full webinar, don't worry, we have recorded it, so it will be available in the future. And please keep us in the loop, because the next year we will have a full focus on in vitro diagnostics in terms of webinars, and we can also support you all along your IVDR transition process and beyond. Thanks a lot, and have a great day. Bye.
Bye, everyone.
Bye. Thank you very much.